Without further ado, I'd like to introduce our speaker today, Dr. Farhia Kabeer. Farhia is currently a postdoc fellow at UBC and BC Cancer Research Center. She works on the mentorship of Dr. Huntsman and Dr. Yvette Drew. She has a clinical background, and she's indeed a well-trained gynecological surgeon in Pakistan before moving to Canada. And she started her cancer research career back to quite a number of years ago at McGill. And initially she was investigating the metastatic potential of breast cancer cells. And during her PhD, she moved to UBC and joined the Sam Parrish's group and continued to study the breast cancer in cancer genomics, focusing on using patient-derived xenografts to study the cancer evolution. After she completed her PhD, she moved on to join the OvCARE and GCI team. Her current research focuses on preclinical models of high-grade serous ovarian cancers, and again using lots of PDX models that she has developed and is developed, and the syngenetic models to evaluate low combinations of target therapy. Her current work is quite vital for developing humanized mouse models and establishing criteria for therapy between conventional and targeted chemotherapies. Hope that will pave the road for future clinical trials. She has received numerous awards that has been recognized as McGill Smith Health Research Award and the CIHR Postdoctoral Fellowship Award. And congratulations, Dr. Kabeer, and I'd like to hear your talk.
Thank you very much for such a kind introduction. First of all, I would like to thank Amira and all the GCI organizers for inviting me here for this talk. My name is Farhia Kabeer. As you know, I'm postdoctoral fellow. I always love to repeat that thing. I'm working with Dr. Huntsman and Dr. Yvette Drew. I'll be talking today on high-grade serous ovarian cancer. So if you look at the title stage, I wrote decoding high-grade serous ovarian cancer. So what do you understand by decoding? Because it's the term usually used for cancers, but this is not the decoding of a computer code, but this refers to the process of uncovering any biological mechanism or molecular characteristics that are responsible for the progression of cancer, behavior of cancer, or understanding the complexity of these cells that are responsible for the cancer to grow, propagate, spread all over the body, and define finally if those cells are going to respond to the treatment or not. So this is actually the term when we use this term decoding for any type of cancer that we. For the sake of today's presentation, I tried to make my slides very small, and I promise I won't go much longer. I will be talking about introduction of cancer decoding, what are the mechanisms we use to decode the cancer, then more specifically on high-grade serous ovarian cancers, its spread, genomic instability, treatment, and what approaches we can try to identify some predictive biomarkers. And today I will only talk about the approaches that we are using in Huntsman lab. And more specifically, the experimental strategies that we are adopting in high-grade serous ovarian cancer research. And I will show you some preliminary results on single-cell data analysis and some patient's plasma analysis that we have so far. And finally, I will be talking about some very interesting models that we are developing in regard to high-grade serous ovarian cancer research that are humanized. Okay, so this is my signature slide. I always like to show it because this gives you the proper insight about the cancers when they grow from the start point to the last time point. So different colors here in this cartoonish picture is representing one clone of the cancer itself. So when I say cancer clone, I mean the group of cells that are related to each other from descent through a unitary origin. So what happens when a cancer or tumor, it grows from one time point to the other time point, these populations or subpopulations, they fluctuate. And as the color shows, some of the populations, they overgrow other populations. And why this happens? Because the cells, cancer cells, they have different fitness advantages, and the cells or population of cells that have more fitness advantage over other populations, they will outgrow the other cells. Okay, this is the normal mechanism for every kind of cancer. So what happens, depending upon the type of cancer, we put the drug in, we treat the cancer with different chemotherapies, conventional or targeted. So what we saw in the clinic, we used to see the tumor shrink down very dramatically. Sometimes we are able to eradicate all the cells. But what happens? The tumor, it comes back after some time. And some patients stay cancer-free for longer time as compared to others, depending upon which type of tumor or grade or stage he or she is having. So what happens when the tumor comes back? If you notice, the colors are changed now. So this is representing the tumor cells, they evolve over time because of these kind of drugs. And now the evolution means there are appearance of new clones, right? It could be possible there are some residual cells that were hiding before, and now they are more fit in the presence of that drug, and they are taking fitness advantage, and they are not responding to the treatment anymore, right? Other than that, the cancer is not only the game of tumor cells, it's also complicated by blood vessels, by the stroma, the cancer. Not only that, then here comes the immune cells. Why I'm mentioning that? Because when we talk about the precision oncology, when we talk about more specific drug treatments, then all these factors should be kept in mind. Number one. Second thing is we need to identify each of these cells, right? We need to target to stop this formation of angiogenesis, like these blood vessels. We need to target the immune cells. Then we need to identify and target the cancer cells. So how can we identify these cells within a tumor? This is another cartoon. I'm now focusing only on the cancer cells. If you look on top on the screen, we need to identify what type of these green cells are, what type of these yellow cells are. So how can we identify? So the first thing is to identify them. It could be DNA for our decoding word back to the word. It could be RNA. It could be the change in the copy number. Okay. So in this way, we can track down the cancer and identify the subpopulation. Then a very important thing is not only one snapshot of the cancer is important, because we need to know what are the changes, what cells are taking advantage in that scenario. So we need to make time series measurement. We need to track them. We need repeated observations of those clones or these cancers while they are time. So while I was doing my PhD, we successfully showed in breast cancers that there are tools available where we can look at single cells and we can actually quantify those cells that are more fit in the growing cancers. So after having that thing, when I started my postdoctoral fellowship, and I was more interested because of my background in high-grade serous ovarian cancers, I wanted to check whether or not in high-grade serous, can we identify these kind of cells, or what are the clonal proportion in those growing cancers, because we know that high-grade serous ovarian cancer is most common and very lethal form of ovarian cancer. And it's characterized by chromosomal instability and a variant copy number. So if you look at the top panel, it's showing the low-grade serous ovarian cancer. It's type 1 ovarian cancer. And if you look on the right-hand side, the copy number of low-grade cancers, as compared to the high-grade, they have high chromosomal instability, and you can see the change in the copy number, right? So it's got a very heterogeneous. And also it gets like the tumor cells in high-grade serous, they don't face any anatomical barrier in the abdominal cavity, okay? And when we diagnose that, it's already spread all over the abdominal cavity. And so what we need to know, especially when we want to treat these kind of cancers, what the main thing that I especially wrote here is we need to know the genetic information on DNA repair path. Okay, because now we, like since a decade, we have very good targeted therapies that are synthetically lethal towards some DNA repair pathways like homologous recombination pathways. And we have seen the patients are quite responsive to those drugs. And that's why these kind of decoding, back to my title of the slide, these things are very important to identify when we are trying to treat the patients. Just a little recap on the treatment strategies in high-grade serous ovarian cancers. Again, platinum compounds, these are taken as a new adjuvant and adjuvant settings along with some targeted therapy. So for the sake of today's talk, I will be focusing on these two because I treated the pediatrics with platinum compounds and PARP inhibitors. So these PARP inhibitors are the targeted chemotherapies that cause synthetic lethality and increase patient survival that are having homologous recombination deficiency in their tumors. Okay, so back to, there are more targeted therapies now. Like in the beginning, I was telling to stop the angiogenesis, like bevacizumab, anti-angiogenic drugs, and then we have immune checkpoint inhibitors that target, there are various target the immune cells and the surface markers on the tumors. So now back to the question, what type of approaches can identify biomarkers that predict treatment resistance or therapeutic response? Main thing, the approaches that we are adopting, don't forget, we need to track the tumor. We need repeated measurements of the tumor. Then only we can originally decode the tumor. One snapshot can give you an idea what the tumor looks like. But if you want to see what changes are occurring over time, you need to track the clonal dynamics. Okay. In patients, yes, we don't want them in patients, the tumors to come back, and we cannot operate the patient again, or sometimes it's not very feasible. But we can take patient's tumor and we can develop patient-derived xenografts, PDX models. What does that mean? You take patient's tumor and put in the mice, and then you can track it down because you can take the tumor and put in the new generation of mice. Okay, and then you can treat the tumors like you want. This is the treatment strategy that we are using right now. Other than that, directly from the patient, what we are doing, and it's relatively less invasive and more feasible to track patient's plasma over time. Before the treatment start, before the surgery, after the surgery, and continue. So these kind of approaches we are using in our lab right now. Let me tell you more in detail what we are doing. So we are taking some specific molecular subtype of high-grade serous ovarian cancer patient's tumor. They belong to BRCA mutant group. They belong to non-BRCA or HRD and HRP, both type of tumors. Then I'm making sure that these patients were never exposed to any kind of chemotherapy before I get the sample from the hospital. So what I do, I take a tumor, I put in the immunodeficient mice, and when the tumor grows, I would treat them either with platinum or with PARP inhibitor, and then I will dissociate the tumor into single cells and see what the tumor looks like in the presence of platinum or in the presence of cisplatin. Similarly, at the same time, I am looking at the clonal composition of patient's original tumor itself, so I can match back what are the differences between patient's original tumor and the xenograft. This is the experimental setting number one that I told you, developing the PDX model. And at the same time, from the same patient, I'm taking the blood before the surgery, and if available, if patient gives the consent to take the blood after the surgery, and looking for tumor burden, copy number changes, and some other biomarkers. So this is the summary of the framework that I'm working on right now. So the first question was like, when I developed the PDX model, first passage one, and then I took when the tumor reached the human endpoint, I took the tumor and put in the next generation. The first question occurred to me was, is it the same tumor that I'm propagating, or the mouse has its own tumor or something else going on before I do some interventions? I confirmed that with histology, and when I looked at the genomic profile, I was quite satisfied that the genomic profile, like if you see chromosome number 3, 7, 8, or 16, 17, the top panel belongs from the patient, and the middle and the last panel, they were keeping the same genomic aberrations similar to the patient's original tumor. So I was quite satisfied. Then I made single cell measurements, and I looked at different clonal compositions. So on this screen right now, you are looking at a heat map where all the libraries for all the single cells that are treated, untreated, treated with olaparib or treated with platinum, all the single cell are put together to create this heat map. So each row is equal to one single cell, telling you the genomic composition of one single cell, and each column is one chromosome. So when we look at different clonal composition, you can clearly see that clone M, it belongs to the treatment groups, and you can see the genome duplication here. So all these color coding belongs to different clones. Clone P and Q, it also falls in one category. So yellow one is olaparib treated, which is a PARP inhibitor. So this was very preliminary first analysis that was done, and it gave me quite a confidence that the things are working, and I'm moving forward with more, accumulating more data and more refining these kind of analysis by collaborating with our bioinformatic partners. So other than these single cell, when I look at, so let me give you an example of this case one. So this case one is HRD case. The tumor high-grade serous was having homologous recombination deficient, and it was actually Yvette's patient. She is still living a very happy life. She's very cooperative. She's willing to give her blood sample multiple times. So she gave her, and the tumor and this data also belongs to this same patient. When I looked at the plasma sample here, I was able to see the tumor content and the changes in the copy number at the plasma level. And when I looked back with the patient's original tumor, the genomic aberrations were same. They were matching with the patient's original tumor. So that means that this was quite consistent what the patient's original tumor was. So this gave me a high level of confidence. Like if I am tracking the patient's plasma down the road, it means like it's much more giving me an idea what the patient's original tumor would look like. And when we take the clonal proportion within the plasma tumor contents, and it came out that the clonal composition in the ctDNA means like this plasma sample were quite similar to the untreated patient sample here. They were more, because this is only one time point, and the other clonal proportion is the time series sample, but the clonal composition type of clone were similar. This is just the preliminary results. We are collaborating very closely with our bioinformatic collaborators to further refine all these analyses by adding more deeper data. Okay, this is very interesting because the same patient, she gave a sample. So Yvette put her on PARP inhibitor, niraparib. She is still taking, and she visited after 6 months back in the clinic, and we asked her like if she is able to give us a sample, and she was very happy, and she said, I can come every week to give you a sample. But we took her other sample and we looked at the tumor contents, and like because she was totally disease-free, her CA125 was very low, all the images are clear, and if you look at the ctDNA, the bottom panel is the follow-up sample from plasma. So we couldn't see much tumor content, but yes, the original level was enough for the threshold bioinformatically to see some of the features confirming like that existed before. So that was again, we were very convinced that the follow-up sample, you cannot see exactly. C3 is almost gone. I cannot appreciate from here, maybe quantification of some numbers, but I cannot. So this is pretty much I have from the PDX, like this, just like the conclusion, like we found some evolving copy number in the PDX model at single cell level, some chromosomes that were mentioned in the heat map, and there were large in amplification, like I just showed you in the plot before, at some specific chromosomes. And yes, I will be doing that with other cases. I'm still waiting for some of the ethics approval from the patient's longitudinal plasma samples, because we also want to identify what is the cutoff value, like when you take patients' follow-up sample. So this is, we are working to for the REP protocol to be approved, where we can take around 5 samples within the first chemo cycle, like because one cycle is for 21 days. There's a gap of 21 days between the 2 cycles. So we are working on that to identify where the tumor level, like in the plasma, it comes starting going down. So so with this, I will just finish the first portion of my talk. If you have any question before I go to the humanized mice.
Thanks. Yeah. So my question is sort of a naive question, I guess, just about the ctDNA detection. Could you more likely to detect a tumor clone that's alive at the time of sampling, or that is perhaps dying?
Yeah, that's a very good.
Spilling its content.
Absolutely, you're very right. That's a very good question. Like even we had the meeting yesterday, we were thinking of those things, like when you say that the tumor content is dying or when it is coming back or something. That's why we are working on the protocol to sample right before the surgery or right before starting the new adjuvant chemo. And then after 1 day, within 48 hours, we will take another sample to compare if the cells are dying or not. Then we will take another sample maybe after 4 to 5 days or something, because chemotherapy is going to go systemic cells will start dying right away. So at least so that we discussed yesterday. David was also there in the meeting, because exactly this occurred to us, like how can we detect the minimal residual disease, for example, or if the disease is coming back, something like that. So yeah, we need to work on that.
Okay, thanks.
Any other questions?
Yeah, with the we looked at. Can you go back a slide to the forward one? So, yeah, this one here. So the little bump on chromosome 1 is probably
Yes.
after.
It is. It's like for sure. It's always there in all the samples.
And so I don't know, since there really aren't any features there which were prominent in the two, because we've lost the main like shows on chromosome 8, chromosome 3, right? That's the most prominent feature. They've gone. And so to argue that anything else is real, we have to say that there's a clone which has lost chromosome 3 and chromosome 8, but is still featuring something else, right?
Yeah, that's true. But I that's the hypothesis. But I am not very confident right now, because still we are developing that computational method, because it could be possible there's no tumor content.
No, I don't think there is too much.
There is
If you go back to all of the slides, so the heat map of all of the DLP, big deal. There you go. So they all have, every clone has a chromosome 3 and chromosome 8, right?
Yeah.
So there's no evidence here that there is a single cell use sample which doesn't have this. So it does. So I think you're close to 0.
Kind of. Yeah, exactly. It means like the drugs are really doing something. Any other question before I move to the
So
next?
one of the things which, I mean, where this would be going is that when patients come in and they want to determine how to treat them, they really want to know whether the patient this tumor is exhibiting features which suggest there's a BRCA mutation, either inherited or something acquired. And what we're wanting to do is to determine whether we can, by looking at the plasma, get that red. So there's a lot of patients who it's very hard to get a tumor sample because they need to be given neoadjuvant therapy. So if you can actually know that ahead of time, even, and the nice thing is that the patient could be in Prince George, it could be anywhere, and they could, blood samples could travel. And then you can make sensible treatment decisions with the knowledge that it's possible when you give the treatment, the tumor may disappear, it may come back, but the problem is the tumor disappears and you can't get that reading. And so we're hoping that this will be sensitive enough for that.
Exactly. This is yeah.
Unlike the pre-treatment sample, the post-treatment sample you showed, where maybe the tumor is almost gone and it's just like a few hundred tumor cells or 1,000 tumors. But if that like decision is being made about neoadjuvant therapy, there's probably like tons of tumors material because the patient will be quite sick at the time, right? And so this could become have a clinical translation pathway, which becomes very interesting and really useful.
Yeah. Thank you very much for adding to that. Okay, let me go to the last few slides of my talk, the humanized mice, bridging the gap of the biomedical research. So far, when I was telling you I'm taking patients' tumor and transplanting them into the mice, those mice are deficient of immune system. Mice immune system, as well as for sure they don't have human immune cells there. So there comes the question, and Brad is here. He was my PhD examiner, and he asked me so many questions on that.
That's a terrible question.
Sorry.
No, that was
Did your psychologist tell you needed to confront your greatest fear again?
no, it was totally a valid question that like made me motivated to work on these models. Like it was very valid. Like how can you characterize a tumor in the absence of like immune cells? Like they are growing in the absence of immune cells, and you are characterizing that tumor. Are you satisfied with that information? Of course, we are not. But this is the only way, because if mice have their own immune system, they will totally reject the graft, right? That's why we are. Then after my defense, I started thinking more about developing these kind of model, and I did some literature research. And when I joined David's lab, I discussed these ideas with him, and he was very supportive, very supportive, and we did a lot of work on that. So when I say humanized, what I mean is the mice that are having circulating human immune cells. Okay. I'm not saying it could be circulating immune cells. It could be we inject some transgenes in them. We make it that are very helpful for expanding some human immune cells. So they are genetically modified when you are defining humanized mice with human genes. It could be tissues or it could be cells. And why we need to do that? So we can understand human biological systems more appropriately, more closer to patients' own dynamics. But it's not very simple to develop these models. I will tell you the story. And the benefits of that, we can study human-specific diseases, immune responses, or nowadays, like very hot topic, immune checkpoint inhibitor drugs that we can test in these models. So Jackie is a master's student in David's lab, and he's working with me in my direct supervision under me to develop all these mice models. So the approach is to take the immunodeficient mice, eradicate them to suppress their own hematopoietic cells, and transplant CD34 positive, sorry, I missed 3 here. It should be CD34 positive cells from the hematopoietic cord blood. And then to biopsy, take the bone marrow biopsy at around 6 weeks and check, are these CD34 cells able to expand human CD45 cells in these mice or not? So the plan was, if we see some engraftment, some expansion of these CD45 mature human immune cells in these mice, then I would put these high-grade serous tumors in them And then we can compare and contrast whether or not there is a difference in these tumors that are growing in the presence of circulating human immune cells as compared to the mice that are the tumors that are growing in the absence of these human immune cells. So Jackie started working on that, but luckily from our collaborators in BC Cancer, Dr. Coney Eve's labs, we had many meetings, many discussions, and she suggested to use ENW mice. E doesn't mean anything. This is just the nomenclature, just the name of those specific breed of the mice that were already injected with some transgenes from human. So the benefit of these cytokines were to promote the development and maturation of human immune cells in mice in a more robust system. Okay, and these mice also contain heterozygous KIT mutation because homozygous were not compatible with life, so it's like heterozygous. So we started using these mice, and another benefit of these mice were we don't have to irradiate these mice. Like here, if you are just taking NRG or NSG mice, you have to irradiate to suppress their system, which is also like mice are not very happy with these irradiation. So using these strain of mice gave us extra benefit of leaving that step. So when we did 4 to 5 or the 6 different experiments, and finally we were able to see, if you look at the last plot, I'm not going to all the gating strategies, but when we look at the FACS plots of the bone marrow cells, finally, after many attempts or everything, we found that the mice, they were exhibiting 70 to 80% of human CD45 cells in the bone marrow. And at the same time, we did like multiple times, and we were able to create like 4 batches of these mice. And we also looked at in the bone marrow of these mice, because the bone marrow was coming from the mice, we also stained that with mouse antibody to see what is the proportion of mice CD45 as compared to the human CD45. And very interesting finding that we found in the 2 mice where it was not stained with human, if you see that these mice cells, they are having very high proportion of CD45, but the other mice that were injected with CD34 means that they do have human CD45, their counter mouse CD45 were very low. Whereas it's just kind of little artifact or doublets, but these mice were never injected with CD34, neither they were looked for that. And then we have some other examples where we saw human CD45 were more than 80% in some of the mice. So that was quite encouraging. And then like so far we are planning to put the 3rd batch of these mice with high-grade serous ovarian cancers. And when we look at different organs and see whether or not on IHC we can see the human seedings of CD45. And if you see on the right 2 panels, they are from humanized mice, where in the bone marrow we saw high infiltration, high expansion of human CD45 cells, whereas the first column is where we did not transplant CD34, and there is no expansion of human CD45. So far, this is my last slide, and I'm just showing you the histology of the high-grade serous tumors that we have successfully grown in non-humanized mice, the top one, and the lower one was the humanized mice that is in the presence of circulating immune cells. So we have harvested already 2 sets of these tumors in NRG where nothing has happened, and 1 set where we have circulating human immune cells, and a set where we have no circulating human immune cells, but the same rate. So we are now looking into these tumors, and we are still waiting for some initial IHC results from these tumors, and whether or not on IHC we can see some tumors, some CD45 positive cells or not. So with that, I would just finish my presentation. So this is from these experiments, like we are able to see circulating human immune cells in these mice. And yes, in the presence of these cells, I saw that at least 3 of our groups, they show up the tumors. Other than the high-grade serous, actually, I'm not mentioning here, but we also we also have endometrial tumors that are growing in the presence of these human immune cells. And we are planning to give them a challenge from immune checkpoint